Ketone bodies are essential molecules when it comes to getting energy for our bodies during periods of prolonged fasting. My name is Alyssa, and in this video we will map out how ketone bodies are synthesized and then broken down within our bodies. We'll start with an overview of ketone bodies during the period of prolonged fasting. We'll then investigate the two processes of ketogenesis and ketolysis, and then finally look specifically at ketolysis within the brain. After watching this video, you will be familiar with how our bodies use ketones for energy when glucose is unavailable. You will be able to answer questions that ask you to recall the steps within each of the two processes studied. First, we'll start with an overview of what will be explored during the video. During prolonged starvation, acetyl-CoA from the beta oxidation of fatty acids can be converted into ketone bodies in the liver. This is done through a process called ketogenesis. These ketone bodies are then transported through the circulatory system to tissues where they can be converted back into acetyl-CoA. The process of turning ketone bodies back into acetyl-CoA is called ketolysis. First, we'll investigate ketogenesis, or the synthesis of ketone bodies. Ketogenesis occurs in the mitochondria of liver cells when excess acetyl-CoA accumulates during a period of prolonged fasting. The enzyme HMG-CoA synthase combines an acetyl-CoA molecule and an acetoacetyl-CoA molecule to synthesize HMG-CoA. HMG-CoA is then broken down by HMG-CoA lyase into acetoacetate and acetyl-CoA. Acetoacetate can break down without the need of enzymes to release CO2 and acetone, which can be transported in the blood. If too much acetone is produced, it can lead to ketoacidosis, a potentially dangerous medical condition in which the pH of the blood decreases. Enzymatically, acetoacetate can be reduced by a molecule of NADH to form 3-hydroxybutyrate and a molecule of NAD+. Both acetoacetate and 3-hydroxybutyrate can then be transported in the blood and used for energy in various tissues. Hydroxybutyrate and acetoacetate are transported in the blood to tissues such as skeletal muscle and the renal cortex, where they are then used in the process of ketolysis. Once 3-hydroxybutyrate reaches the mitochondria of these cells, it is oxidized to acetoacetate via the conversion of NAD+ back to NADH. The enzyme thiophorase then converts acetoacetate into acetoacetyl-CoA. Thiophorase is only present in the tissues outside of the liver so that it cannot catabolize the ketone bodies that it produces. Acetoacetyl-CoA is then converted into acetyl-CoA, which is then used in the citric acid cycle for energy. If the fasting period extends beyond 1 week, the concentration of ketone bodies in the blood becomes high enough for the brain to begin metabolizing them as well. During times in which food is plentiful, the brain derives its energy primarily from glycolysis. Glucose is converted into acetyl-CoA via a multiple-step process which requires pyruvate dehydrogenase. During periods of prolonged fasting, pyruvate dehydrogenase is inhibited in the brain to prevent glycolysis in favor of ketolysis. This is done so that the proteins essential to the function of our bodies are not broken down to eventually be used in the process of glycolysis. Now that we understand the importance of ketone bodies during a period of prolonged fasting, let's have a look at some practice questions to help solidify this knowledge. This question asks you to recall during which phase the thiophorase enzyme is present, and therefore, where this enzyme acts. The answer is B. Thiophorase is one of the enzymes responsible for converting ketone bodies into acetyl-CoA. This process occurs in both the brain and the renal cortex, but remember that ketolysis only occurs in the brain after 1 week of fasting, and not 48 hours like the question stem prompts, so answer C is incorrect. Therefore, the only match is answer choice B. Remember, the liver does not contain this enzyme essential to ketolysis, and this process does not occur in the bloodstream, eliminating both answer choices A and D respectively. This second question is asking you to think about the main molecules present during a period of prolonged fasting. The answer is C. During periods of starvation, glucose is not readily available due to decreased glycogenolysis and gluconeogenesis, eliminating answer choice A. Acetyl-CoA and thiophorase do not exist in the bloodstream, but in the mitochondria, eliminating answer choices B and D as well. This leaves answer choice C as the correct answer. Acetoacetate, a ketone body that is transported in the bloodstream, would be expected to be present at relatively high concentrations during periods of starvation. Excellent work if you got those questions correct. In this video, we looked at the process of ketogenesis, the synthesis of ketone bodies from acetyl-CoA. We also investigated the process of ketolysis, the breakdown of ketone bodies back into acetyl-CoA. Finally, we explored how after 1 week of fasting, glycolysis within the brain lessens and ketolysis increases to spare the important proteins within the body. Thank you for watching this video, and good luck studying for your MCAT.